Good morning, everyone, and thank you for attending Chardan's 8th Annual Genetic Medicines Manufacturing Summit. My name is Daniel Gataulin. I'm one of the senior research analysts here at Chardan covering biotech. It is my pleasure to introduce our next guest, Craig Malzahn, SVP Technical Operations from REGENXBIO. Craig, welcome.
Thank you very much.
So the format for this session is roughly 25-minute fireside chat. If audience members would like to ask a question during the session, please type it into the question box under the video player, and I'll make sure to ask it on your behalf. With that, Craig, let's go ahead and get started. So the first question, if you could please provide a quick overview of REGENXBIO and where its manufacturing process currently stands and how it evolved over time.
Sure, and maybe I'll start a little bit with this quick intro video on our, I still call it a relatively new manufacturing facility. So
Sounds good.
go ahead and play. This is the REGENXBIO manufacturing facility that we opened for operation in the summer of 2022. The REGENXBIO Manufacturing Innovation Center is a GMP gene therapy facility designed and built for the manufacturing of NAV technology-based AAV vectors at both clinical and commercial scale, supplying product for both large market and rare diseases. The state-of-the-art facility in Rockville, Maryland is housed in the same building as our research, process development, and clinical teams, which allows us to accelerate the process of developing AAV therapeutics. The facility includes 2 independent bulk drug substance production suites, a drug product fill line, and quality control labs. The process development capabilities ensure we are continuously innovating and leading the AAV field in advanced manufacturing technologies. So that's just a brief intro there to our new facility.
Excellent. Thank you very much, Craig.
And then maybe I'll take your question then.
Yes.
So I think you asked about how our manufacturing process has, where it is today, how it's evolved over time. Really, we've been in the business of drug development since 2015 or so. Probably similar to a lot of gene therapy companies. We started on an adherent or hyperstack manufacturing platform, which is really technology that was developed more so from an academic lab type of environment. Process there, it produced quality product, but it's quite inefficient, very manual, inconsistent operationally, and not the easiest to scale. So we invested considerable time in adapting that HEK 293 cell line to grow in suspension. We selected the highest producing cell clones. We optimized triple transfection process, and now cells make more capsid. They package more transgene more efficiently. It also allows us to streamline the scale up of the purification process. We've optimized the formulation, so now we've got stable capsid, long shelf life. All of this investment in the innovation here in the process has brought us to what we call today as our NAV Express platform process. It uses a proprietary NAV Excel production cell line, and it's proven a platform that's robust across different capsid serotypes, different transgenes, and really it's an industry leading productivity system. Titers are industry leading, and our production yield is really come a long way, really remarkable from the original processes with nearly an 800-fold productivity increase over the evolution of the process.
Got it. Thank you for that overview. Some companies rely on CDMOs for their manufacturing needs, while others invest into in-house manufacturing early on. What were some of the key factors that led to REGENXBIO deciding to take on its own manufacturing?
Yeah, we manufactured at CDMOs for really years prior to building our own manufacturing facility. When you're just getting started, it makes sense. You're able to sort of avoid capital spend, minimize some of the fixed operating costs. And you have some flexibility when it comes to process changes and that sort of thing. I would say really confidence in our NavExpress suspension-based platform, defining that process, being able to apply that across our entire portfolio was one of the key enablers. Clearly a maturing pipeline of multiple programs, and then really a desire to control our own destiny from a manufacturing perspective, from a quality, schedule, cost perspective, led us all to building in-house capabilities. And we've seen a lot of benefits from this, already. It gives us really, really strong manufacturing schedule flexibility. We can add batches quickly. We can remove batches from campaigns. We can switch over from one product to the next quickly. We can reprioritize where we need to reprioritize around programs. And then from a technology basis, we're able to accelerate new technologies quicker into the plant, make those process changes, bring forward GMP batches. Having it under the same roof as our process development team also allows us to troubleshoot problems, really real time on the floor. Oftentimes, we're even able to save a batch because we've been able to make a quick scientific decision to correct an issue that we might be seeing in a GMP process. And overall, it definitely has reduced the supply cycle time to the clinic. Being able to manufacture drug substance and drug product in-house really enables a faster supply to the clinic.
Got it. Thank you. And for manufacturing in the gene therapy space, especially when you scale to larger indications, it is quite important, if not key, to make the process more efficient. So you mentioned some of the things that have evolved in your manufacturing capabilities. But maybe you can spend a few more minutes to describe in more detail some of the key considerations to the manufacturing process that you have implemented to make it more efficient.
Yeah, I would say I would start with, and I think we've seen the biggest impact in efficiency gains coming from cell line development. So cells that produce more capsid per cell than others, really investing in higher producing cell clones has driven our titers up considerably. We also run a triple transfection process with our HEK293 platform, and we've done optimization around those plasmids for better transduction, more efficient transfection. We've seen significant gains there in productivity with more optimized plasmids. Certainly downstream, a lot of the focus is around reliability and making sure that we can produce the same quality profile each and every time. But we've gotten a lot better, and I think the industry has gotten a lot better in separation of full and empty capsids. We use ion exchange chromatography system for doing that, which is a scalable technology that's definitely helped us move from smaller scale up to full production scale. And then there's little things like in some of our programs where it's rare disease indication, maybe we're not making a high volume per batch, just really optimizing sample volumes, reducing waste downstream has all benefited us quite a bit with efficiency. And then the last one I'd mention is just being able to demonstrate. Now we've demonstrated we can operate at 2,000 liter scale. Our process is scalable up to that level. Larger batches you gain economies of scale if you have to go to that size bioreactor. Whereas you're able to primarily keep your labor and your overhead costs fairly flat when you go from 200, 500 to 2,000 liter scale. It provides a really nice advantage for certain indications that need it.
Got it. So I wanted to talk a little bit more about costs, and Regenx mentioned previously, especially for some of the programs where high doses of systemic therapy are required, such as DMD, potential advantages in COGS versus some of the competitors. So can you talk about specific innovations in your strategy that you're looking at to potentially reduce or circumvent some of the key cost drivers?
Yeah, absolutely. Our platform today, we're very happy with the triple transfection platform for HEK. As I mentioned, we're continuing to develop different plasmid systems and optimizing around that to reduce cost, really with a goal of producing more per cell. And then we're interested longer term in innovations such as stable cell lines. There's some promising systems out there that we continue to take a look at. I wouldn't say these are must-haves for our current programs by any means. I think we're quite happy with the NavXpress platform technology that we're using. We continue to optimize around the bioreactor conditions, the nutrients, optimizing enhancers for transfection, looking at new complexation reagents, more scalable purification methods, improving those, better affinity resins, more robust ion exchange matrices. All of these are areas that we continue to invest in, look for new innovations, make sure we're staying at the front of the industry in regards to that. And all of those are going to help us continue to be more efficient, more effective in our costs.
Got it. Thank you. And so you talked a little bit about your manufacturing facility. It went online. Can you summarize its capabilities in terms of capacity limits, and maybe to put it in perspective, your two programs that you're currently or three programs that you're currently developing actively, RGX-314, how many doses of that can be produced from a single manufacturing run? In comparison, maybe how many doses of RGX-202 for your DMD program can be produced from a single manufacturing run? And will all that be sufficient to meet the patient demand?
Yeah. So we, as mentioned in the opening, we opened our GMP manufacturing facility in the summer of 2022. So we're coming up on a couple of years of operation now. The facility has two independent drug substance manufacturing suites. So we're able to manufacture multiple products simultaneously with parallel processing. We have a drug product suite as well for liquid vial filling. And the majority of our quality control tests are also done within our facility in our quality control laboratories. The facility itself is designed to meet both our clinical demand and global commercial demand forecasts for our lead programs that we have today. The facility overall, we did actually get feedback from the regulatory agencies prior to finalizing the construction plan. So I think we have confidence that this is going to be a commercially viable facility. From a capacity standpoint, we are quite flexible. We are able to operate either at 500-liter bioreactor scale or up to 2,000-liter bioreactor scale. So that gives us, really a fourfold capacity swing there. And I also mentioned we've now successfully executed process validation campaign through the facility. So we're really in those sort of final stages of getting ready for commercialization. The drug substance capacity that you asked about for RGX-314 and RGX-202, we've got capacity in the facility. It's designed to be able to meet our commercial demand forecast for the U.S., for global markets as well. A little bit of context for that, of course, is RGX-314. It's an ocular program. So obviously with ocular, you're talking about quite a small dose per patient. So a single batch is going to make you thousands or maybe a couple tens of thousands of doses per run. So you're quite efficient with that type of program because of the the small organ of the eye and the small dose. Whereas in contrast to 202, it's a muscle indication, so it's a large dose per patient. Obviously, we've not selected our final dose that we'll move forward with, but I would expect us to be in whatever dose that we choose for our Duchenne program, that we're able to make, tens, 20s, 30s of doses per run to meet a rare disease type of indication.
Got it. Thank you. That's very helpful. And in terms of versatility, so you mentioned you can run a couple of runs in parallel. So to maybe expand on versatility, how quickly can you transition from manufacturing a product for one indication to manufacturing a product for another indication, or how many parallel runs can you accommodate?
Yeah, great question. So when both production trains are operating, obviously we can make two products simultaneously there. And the facility is designed in such a way where we've segregated sort of upstream seed expansion operations from production harvest and from purification. So we're actually able to execute rolling product changeovers, which also gives us some efficiency in moving from one product to the next. So, I think really it's a matter of a few weeks to move from one program to the next. Some of that will depend on the complexity of the changes from one process to the next. That is an advantage being on a singular platform, where, moving from one program to the next is really usually down to relatively small changes. But to answer your question, I mean, it's really just a matter of 2 to 3 weeks. You can move from one established program to the next.
Got it. Makes sense. And so for RGX-121, you're on track to submit a BLA later this year. So I understand you have ongoing CMC interactions with the FDA. Wanted to ask if you can comment on those interactions to date. Any concerns or any praise that you've received from the FDA?
Yeah. So RGX-121, right? Small rare disease indication for MPS II, Hunter syndrome. We've had a lot of interaction with the agency over the last few years. In particular, we are pursuing an accelerated approval pathway for that program, and I would say, FDA has been quite supportive of that type of an approach. And they've been, I would say, very supportive in acknowledging the CMC challenges, in particular when it comes to, rare disease or ultra rare disease indications. So they've been open to creative approaches to be more efficient, while making sure that we're meeting the compliance and the high quality standards for a gene therapy product like ours. So I would say, things like flexibility in the process characterization space, where we're able to leverage data from one program to the next when it's on the same manufacturing platform. Creative ways to approach process validations or minimizing product waste. It's very expensive to make these programs. We want to maximize the amount of material that we're able to make and then deliver to patients. So things like reducing sampling or staging process validation so that you're not, forced into a situation where you make batches and it expires before you can use them. So I'd say the agency's been very open to those approaches, along with surrogate studies in the CMC space. And then ultimately, we'll see soon enough through the BLA interaction with the agency once our filing goes in. where are we on the same page with things? But I would say overall, the dynamic has been, very positive with the agency.
Okay, understand. And so I understand, there are some CMC questions that are unique to this program, but there are some overlapping CMC items that would be relevant to your 314 and to your 202 programs as well. So wanted to ask how you think the experience with 121 BLA submission and those interactions will help you to potentially streamline the process for the other programs.
Oh, it's going to be huge. There's no doubt. Going through our first now BLA filing, I think there's, it's an invaluable learning to go through to put forward that data set, better understand what the agency is, most concerned with, where we're hitting it out of the ballpark and things are looking really good. That's helpful too. All of that information that we'll learn, the dynamic back and forth with the agency, we're set up in such a way with 314 and 202 from a timing perspective to be able to take a lot of that information, incorporate it back into those programs, in particular in the CMC space, and really de-risk 314 and 202 as we go towards BLA submission, over the next couple of years with those programs. Really in gene therapy, I mean, from a manufacturing perspective, in general, you make so few batches, GMP batches, that when you're able to sort of pull learnings from one program, whether it's agency feedback or your own experience, and you take that knowledge and can apply it across the platform, across programs, there's no question it gives us a big advantage.
Got it. I have a couple of more general questions, but before I ask, I want to remind the audience, if you have any questions, please feel free to type them in, and I'll make sure to ask them on your behalf. I guess a more general question that I have is, from your perspective, what do you believe are the biggest CMC challenges in gene therapy manufacturing are currently?
Yeah, I mean, I think this is still a maturing industry, definitely from a manufacturing perspective. So being able to reliably produce a high quality product from a drug supply standpoint, and staying off of, it's always, when you're living in the CMC space, you always want to stay off a critical path on your drug development programs. I think that's still a challenge. Big challenge for CMC is to be able to produce reliably, consistently, to meet the demands of the clinic, and then ultimately through commercialization. I think a big part of that, it's not just in-house GMP manufacturing, but it's all the other support groups associated with that, the quality control teams, the analytical development teams. Just as important as making product in the manufacturing space is being able to test it reliably and feeling confident about that as well. So that's an area that REGENXBIO has invested in very early, is those analytical method capabilities, so that when we make a batch, we know it's hitting the quality attributes that we're after.
Got it. And so in the past, I guess, couple of years, we've seen some of the disruptions to supply chain in manufacturing in general, but in gene and cell therapy in particular. What steps have or has REGENXBIO taken to minimize the impact on the supply chain?
Yeah, good question. I mean, it's, gene therapy manufacturing, AAV, viral vector manufacturing, we're primarily using single-use systems, a little bit different than, maybe traditional biologics, biopharm, mAbs, that type of thing, that are using a lot of stainless steel reactors and facilities. So what that does for us is it means that our suppliers are even that much more important to being able to to manufacture on time, to execute well. So to really manage our supplier base, our approach is to validate in really as many multiple sources to the process wherever we can. So we have optionality. We can go to one vendor, we could go to the next vendor. If one vendor is stocked out, we could shift over to somebody else. So we do that wherever it's possible. Not every instance that is possible. In those other cases where we might be single sourced, then we're going to carry safety stock inventory. And again, this is another advantage to having a platform process. You can carry safety stock that's common amongst multiple products, and you're much more efficient with that. You don't have to carry as much inventory. You don't have as much obsolete inventory as you go. And then we've got a program, supplier relationship management program, where we've identified our key strategic suppliers. We interact with them frequently on a routine basis. We take a look at performance metrics, quality, delivery on time, things of that nature, and make sure those relationships are strong so that when we, maybe we run into a problem or we need something expedited through, we've got a relationship there. We can move quickly to get a resupply. I would say in addition to that, where we can with our facility, we've built in some redundancies for ourselves in-house. So we can do things like, we make our own buffers and media solutions, and we're able to assemble our own tubing sets and kits and things of that nature, as well as buy them in from suppliers. So that provides some risk mitigation as well from a supply chain standpoint.
Got it. Thank you. And one last question, maybe in 30 or 60 seconds, if you can summarize, the benefits and what differentiate REGENXBIO from other programs, and maybe mention a thing or two that you believe investors are not fully appreciating about the REGENXBIO story.
Yeah, thanks for that final question. I mean, REGENXBIO has come an incredibly long way. We've built internal end-to-end capabilities for discovery, research, drug development, and now GMP manufacturing. So that gives us a great deal of flexibility and capability to execute programs from early stage through IND, through our clinical phases, and then to commercialize these programs. So it's quite an exhaustive build around end-to-end capabilities. I'd also mention we've just recently dosed over 1,000 patients now with gene therapy through our programs. That's a large data set of clinical experience across our programs that we're really, really proud of. And of course, we've got clinical experience, numerous studies, up to a dozen studies or so across multiple therapeutic areas that we've executed in ocular, in the CNS, central nervous system space, in muscle tissues. So that's a broad range of experience that we've gained. And then I'd say, just to end it, it's really the combination of some fantastic people that we have here, the great science that we do, and having capital in place to position us well for commercialization for both large indications and rare disease indications as well.
Got it. Then thank you very much, Craig. That brings us to the end of our session. I really appreciate the time and look forward to connecting with you again. Thank you.
Thank you very much.